Good morning and good evening, everyone. This is a new version of Healthcast, the Center for Management of Health Services at IIM Ahmedabad, the podcast we have launched this year. Today, we have with us Dr. Mehta, who is the Chief Medical Officer at the Albright Stonebridge Group and a faculty at Harvard Medical International. Also, he is affiliated with a lot of his past scholarly work with the LV Prasad Eye Institute and the Wellcome Trust funded center over there. So thank you, Mehul, for joining us in this conversation.
Thank you, Chirantan.
Today, Mehul, we are going to chat a little bit about the future of clinical trials and COVID-19 beyond what were we learning from COVID-19. So just some opening remarks from you. I mean, there's a lot of conversation that has been happening since the first Pfizer vaccine released data and got approval in UK, and now we have the India approvals as well. Country by country. Before that, we saw Sinovac approvals and Gamalaya approvals. And even before that, pharmaceutical productivity and R&D enthusiasts were talking about how clinical trials may be a big drag to pharmaceutical innovation and productivity. So if you have some opening thoughts on what are you observing and what your general reaction is to the future of clinical trials post the pandemic.
Yeah. Yeah, sure. Thanks for first inviting me and speaking to your students and your audience. I think if you just step back, look what happened in the last 1 year. We had a pandemic. We didn't know it was a novel pathogen. And literally, we pivoted to having a vaccine out in a year, right? Now, this is unprecedented in terms of what it's taken to build vaccines in the past. Uh, and then, if you see why this happened and what are the lessons we can learn, I think that forms a very interesting uh, case study. This whole
Mhm.
pandemic is a case study in terms of how do you take something as fundamental as a new pathogen and figure out how you're going to treat it because you have to now treat the world for it, and how can you create the entire process of discovery to translating that to the bedside, not really actually uh, cutting the corners of the main components that go into it, but optimizing the whole process in a very, very, very compressed timeline and get out something that we know from all the data that's coming out has effectivity. It crosses the 50% threshold and benchmark. So I think this sets up a very interesting discussion on what can we learn. Because one thing I just say, if I really have to step back, is the pandemic has forced us to rethink everything,
Yeah.
and it has forced us to rethink everything simply because we had to respond. And
Yeah.
a lot of the ways we approached things in medicine, in regulation, in discovery, in application, we had to throw out of the window.
Hmm.
And I think a lot of those are not coming back. Many will come back, and even if they will come back, they'll come back in a very different way. So yes, it is a new paradigm and
Hmm.
it is a new normal. But the question is, do we want to rest at a normal, or how can we use this to continuously improve and optimize, but do it in a manner that's safe and effective?
Okay. Um, so one of the old normals came from the thalidomide scandal, and the FDA tightened up, at least in the
Mhm.
US. And from then, you would say, here comes another shock. Let's say the medicine regulatory system has faced, and it has shown its own uh, compromises, I would say, on science versus effectiveness versus safety. And now we have these vaccines. Some are 90% plus, some are greater than 50% plus, the WHO specified standard. And along the way, you also have these Indian versions of vaccines that have been launched, which are called as emergency restricted emergency restricted
Use
approval in clinical trial mode, which is
EUAs, right?
a new language that the regulator locally is using. Also, we know along the way what has happened in these vaccine cohorts is the old vaccine makers like Sanofi or Merck have actually played cautious. I mean, if you have been following the innovation race here, they are taking it slow at the same time. And what does this all mean for patients? How should patients think about vaccines and taking vaccines. Just before I started this podcast, I got a message from a friend in the US saying, "Hey, I'm not going to take even the Pfizer vaccine because I have allergies and my kids have allergies." And here in India, we are having this debate on how safe is Covaxin, etc., etc. So, how should the patient think about it? If you can help
Yeah,
with some
sure.
algorithm on this
Yeah,
process, yeah.
yeah. So I think there are lots of things you've asked, and let me unpack it and make it simple. I'll simplify it for you. So let's take it step by step. First of all, the vaccines that emerged in 1 year, what happened to make them emerge, right? And the thinking is, oh my God, it was 1 year compression of time. Normally it's 15 years for a vaccine. There must be something that was done that is actually not thought through. So let's really think about that. In the past, when there was a epidemic of an infectious disease, and one had to develop a vaccine of a novel pathogen, the situation was the following. Most of the epidemics of infectious diseases started in low and middle income countries, right?
Mm-hmm.
The cohort of patients or population affected by the epidemic is limited.
Mm-hmm.
And frankly, vaccines as a prophylaxis is not a money making space. It is really
Mm-hmm.
a public health space. So now, in the past, you had a small population in a part of the world that is emerging economically, that was affected. And really, there's not a commercial drive to go. So you had to have WHO or Gavi or the Gates Foundation or these multilateral institutions to say, let's fund it and do it.
Mm-hmm.
Now, what happens there is that funding is limited. Anyway, there's a concern going into vaccines. There are few manufacturers that want to do it. So everything becomes linear. You first have to figure out, oh my God, what is the pathogen? How does it act? What is its biology? What is its structure? Create animal models. Then you take the animal models and you look at then what can you do in the animal models to tackle it. Then you go through the process of doing, tackling it with certain types of vaccines that you start developing. That whole process takes many years. Then you go through phase one, then you go through phase two, phase three, then you get approval. And assuming somebody funds all this, then you get some manufacturer who's only after they see the results will manufacture and then you distribute.
Right.
Now, your cohorts of patients are limited, so your phase one takes time, your phase two takes time, and the people vested in this are limited. You have a pandemic. The whole world is your cohort.
Mm-hmm.
The whole world is vested. So, what has happened is resources from everywhere have been thrown in.
Mm-hmm.
That's the first thing to remember. So, when you're taking something that is so limited to something that's global, you're going to actually see speed, right?
Mm-hmm.
Because it's all, every single lab that I know of who could pivot it to look at COVID, every single lab.
Mm-hmm.
Right? Look at the power of that. The second thing is that once we knew this was a coronavirus in
Mm-hmm.
January, and that's when the
The desert.
Chinese from Wuhan put the whole genomics up, right? We knew we had seen this before because
Mm-hmm.
we had seen SARS and MERS.
Mm-hmm.
So all the development work that had been done in terms of biology and all, this was the first cousin, right? It's just changed its spike, it's changed some of the areas that the way it attacks the cells. So all that work that was done on the animals immediately came into use. So you
Mm-hmm.
cut down that entire time.
Mm-hmm.
Suddenly you were able to say, we had actually even looked at things for an RNA virus. We had actually looked at things with Ebola also. So let's pull from there. So the work that we had done, SARS, MERS, Ebola, there's significant drivers to speed up the initial, what is the vaccine strategy process, right? There were people who were working on, let's look at Oxford. They were working on looking at malaria vaccination, right?
Right.
They had
Right.
already created the platform. They had already tested it in patients.
Right.
So you see people who pivoted
Mm-hmm.
it's not a question of why did Pfizer pivot and why did Merck not pivot? Who had the ability, based on the work they had done, to really pivot fast?
Mm-hmm.
And so Oxford could pivot. Bio and tech, because the work on mRNA that was being done for many years, first at UPenn by this brilliant scientist who really was working, and at that stage, she was pushing it while nobody was really paying attention to her, she realized that this is a terrific way to deliver vaccines. So we'll
Right.
talk of platforms in a second. So let's talk of timing. So now you have this speed because you have available science that's ready, and everybody who had available science took the lead in the race. Other people wanted to enter, but they are much late earlier because they had to develop things themselves. Now you go through the phases: phase one, phase two, phase three. Populations to do vaccination with, populations to really tackle huge numbers, right? Because the pandemic's exploding. It's not that you have a localized epidemic in one part of the world or a
Mm,
localized spread, huge numbers. Then we know that phase one, post animal, phase one is under 100, and phase two is a couple of hundred, and then phase three goes to tens of thousands. You're actually can compress phase one and phase two. So you
mhm,
can overlap it. You're not saying I'm not doing phase one, but you can say phase one and phase two is the cohort numbers just increase. You're looking at the same thing, safety, effectivity, response, and trying to get your dosing right. Phase three is still the same cohort. Say phase three, you're doing tens of thousands. So now you overlap. And then the most important thing that happened is manufacturing became at risk.
mhm,
You're not saying I want all the data to come out and then I'm going to figure out and then I'm going to manufacture, but you're saying manufacturing can become at risk. That's
mhm,
what Serum Institute of India did. That's what
mhm,
many people did. We are going to invest. If this vaccine doesn't work out, well, it's more important to invest this kind of money now so
mhm,
that we can respond to the pandemic. So you scale up manufacturing. So people were ready, right?
mhm,
That's what compressed the timeline from a development cycle perspective. Obviously, what happened on the authority side, they started to give emergency approvals. That means they say we are still looking at data, but we see enough data that allows us to say there's an emergency approval, not a full approval. Some vaccines have been fully approved, like, you know, China and Russia decided to go to full
right,
approvals on their
right,
vaccines, and that's a separate story. But let's talk about how it is done for the main vaccines we are looking at least the ones you're talking about. So that's how this came about. Now, all vaccine platforms are not equal.
mm, mm.
And the second part of your question was, built into that the concern that are we looking at and how clinical trials were done. I think there were two parts in it. How patients were recruited.
Can I be a little more specific and help you here? I
Yeah, yeah,
think that may help you. So the UK regulator, MHRA, used the UK arm of the AstraZeneca trials, if I'm not wrong, and used
yeah.
that data to approve. The
Yeah,
Brazil arm protocol was violated and that was not used as data. But in India, we don't know which arm's data has
mm,
been used
yeah.
for
You're right.
even Shield approval. So,
Mhm,
and the other sociological story here is India has never historically had a vaccine hesitant crowd. People
mhm,
have, like, if you look at smallpox in the history of
mhm,
smallpox vaccination, people have as a society adopted vaccines, unlike, let's say, in the US. But with this process of approval, where we don't know what data is being used, would we create a false sense of, at one end, hesitancy, and at the other end, a false sense of, okay, all is okay, and that could create the risk of a spike again tomorrow for the virus? That's my concern.
yeah, I understand. So, okay, to answer your concern, the question is what data did they use? Let's step
Hmm,
back and say what are these platforms? Because there's
hmm,
confusion about the platforms. The simple way to look at platforms is they bucket themselves into three platforms.
mhm,
The first is a platform that uses the virus or parts of the virus made outside. So you
mhm.
take the virus, you kill it, which is the Bharat Biotech ICMR approach, which is a established technology. We've done it for other, pandemics. I
Zika,
mean,
yeah,
we've done it. We kill the virus. We know how to do it. We give it with adjuvants because you have to give it with adjuvants. So that's the first. Let's talk of data in a second. The second technology, platform, is to say, why do I give even parts of a virus? Can I avoid it? Can I find which part of this virus is the most important to create an immune response against?
right,
And if we create an immune response, we can actually block it from entering the cell.
right.
It turns out to be the spike.
Hmm,
The spike, because it's a coronavirus, it has all these spikes, looks like a crown, it's called corona. The spike is critical to enter the cell because that's the key that opens this receptor in the cell called ACE2 to enter. Now, the question was, can you actually have the cell? because our cells produce protein all the time, right?
Mm-hmm.
It's a constant process. So can you instruct the protein production mechanism in the cell to produce the spike?
Mm-hmm.
Now your cell produces a spike. It's a very elegant way to do it, because you don't have to give it outside, kill the virus, adjuvinate it, and all that. And if there was any concerns in the past, they were around adjuvants. What are the adjuvants you're putting in? Aluminum was, they say culpable for certain conditions.
What is the
They
role of
had,
an adjuvant for my MBA students? If you can just
so an
clarify.
adjuvant. So when you give a vaccine, let's say you kill the virus and give it, the amount of antigenic exposure that you're giving to the body is limited by how much you can vaccinate, right?
Mm-hmm.
So in order to boost the immune response, you you connect it, or you give it with certain chemicals or
Mm-hmm.
substances that can create an inflammatory immune response. So
Aha.
it's like turbo boosting it, right? Because you're limited to the dosing you can give from outside. You're giving a live virus, or you're giving a weakened virus.
Right.
You can create parts of the virus outside and give it, which is called recombinant technology.
Mm-hmm.
You need an adjuvant. Now, adjuvants, remember, our immune system has two parts to it. Very simply put, one is the antibodies and one is the cell-based immunity. They are
Right.
both equally important. Antibodies is more immediate, cell-based immunity lasts much longer.
Right.
Now, adjuvants want to boost that.
Mm-hmm.
They want to boost that. Therefore, you have to mix them, because some adjuvants create more of antibody response, some create more of cell-based response.
Mm-hmm.
You use the body to produce parts of the virus, and the most important being the part, you don't need to adjuvinate it, because you are actually using the body's own mechanism, and multiple cells are producing internal to the body. Now,
Okay.
how do you make the body cell produce it? Well, you have to instruct the cells produce
Mm-hmm.
this particular type of protein or spike.
Mm-hmm. Mm-hmm.
How do you instruct it? Well, you can actually use another virus,
Mm-hmm.
change its DNA
Mm-hmm.
with the DNA sequence you want. That virus goes into the body, it infects the cell just like it would, it releases the DNA. The DNA goes into the cell's nucleus, and it instructs the cell's protein mechanism through messenger RNA. So the intermediate between that is the messenger RNA. Messenger RNA is just taking the message. It has the sequences for the protein production part of the cell
Mm-hmm.
to produce the spike. Okay.
Mm-hmm.
That is the basis of many, many vaccine platforms. Oxford is AstraZeneca is a virus. It's an adenovirus.
Mm-hmm.
That is the carrier, right?
Mm-hmm.
Johnson and Johnson is an adenovirus.
Mm-hmm.
Russia is an adenovirus.
Mm-hmm.
Sinovac is an adenovirus.
Okay.
Different adenoviruses.
Mm.
Now, when you go down that path,
But this is not the mRNA platform that you are talking of.
no, I'll come
Okay.
to mRNA. The third
Okay.
is mRNA.
Okay.
So it's important when you think of all this, you think of what you're getting. It's like saying I want transportation, right?
Mm. Mm.
Now, are you taking a bus? Are you
Mm.
taking a car? Are
Mm.
you taking a ship? They're
Mm.
all transportation. Are you taking
Mm.
a bicycle?
Mm.
So broadly, this is the way to divide it. I'm giving a simple framework to
Yeah.
divide.
Yeah.
So
Yeah.
let's just talk of the vectored vaccines. This adenovirus and all come under vectored vaccines. You're taking a vector to
Mm-hmm.
deliver the sequence. So what's the good news about this? You're not delivering the virus. You're delivering a code.
Right.
The other good news is you're going into the body and the body can amplify this. So you're
Right.
not giving any adjuvants, but you're using a vector. Now, when the body's immune system looks at this vector, it's going to say this is an infection in and of itself. It's a virus.
Right.
So it will react against the vector, and therefore you have the problem of vector immunity.
Okay.
So if you have been affected by the virus before,
Mm-hmm.
then chances are that when the virus is injected, the vaccine is injected with that vector, the body's immune system will kill the carrier before it can do its job.
Okay.
So the choice of adenovirus you choose has many things to take into account. Is it safe? Is it been used before? Because it goes to the nucleus, you have to worry about those things. It's DNA virus, it's an adenovirus. So Oxford decided to choose a chimpanzee adenovirus. Why?
Right.
because in their study in Africa, when they were doing it for malaria, they found only 1% of the population had immunity against that
Mhm.
vector. If you look at what Johnson and Johnson have done, they've chosen adenovirus 26
Mm.
because that's less common to infect humans than adenovirus 5. And what
And
Russia
actually, yeah, go ahead.
and what Russia did is they said that anyway, if you understand immune response, you have to give the first dose and then you have to give the second dose to get a good boost response. So
Mm.
you're giving
Mm.
2 doses. Russia said if we give the same adenovirus twice, the second time we give it, the immune system will be primed and will attack it. So we'll give two different adenoviruses. So they
Okay.
start with 26 and they go to 5. That's the
Okay.
sequence Russia has, right? So that's vectors and vector immunity. Now, could you
Mm.
use some other virus? Yeah, people are looking and saying, could we use rabies virus? And that's all happening. That's very early development, right?
Yep.
Could we use some other way to do this? So that's the vector. The third is mRNA.
Mm.
Now, mRNA, what is mRNA? All our cells have messenger RNA. M stands for messenger. Messenger RNA takes the instructions from the DNA from the cell's nucleus through the cytoplasm to the protein production mechanism, gives it to the protein production mechanism, and then after a certain period of time, 36 hours, is destroyed by the cell's own mechanism. That's what messenger RNA does constantly. So this scientist, Hungarian lady scientist in Penn, who came to Penn, said, why can't I use messenger RNA to instruct the body to produce proteins to treat disease,
Okay.
right? Now, the way you do that, so if you have these proteins that your body needs, certain protein types, it can be produced. If I see a cancer cell and I actually know that the structure of the cancer cell is X,
Mm-hmm.
certain structure, I can get the body's cell's protein mechanism instructed by the mRNA to produce that protein. It gets produced in the cell, it comes out of the immune system recognizes it and says, oh my God, this is foreign, we have to attack it. It scans the body, finds these cancer cells and destroys them. That is immunotherapy. That's a form of immunotherapy.
That's why they are saying the COVID-19 mRNA vaccines can also solve cancer potentially going
Well,
forward.
it's a platform to do lots of things.
Yeah.
It's
Yeah.
a very novel platform to deliver. Now, so these guys who are already doing it for cancer said, if I can produce a protein for cancer, why can't I produce a part of the coronavirus, the
Mm-hmm.
spike? And that's exactly what the mRNA is doing. It's coded for the spike. Now, the question is, what has made it, why did it not occur before? Because when you inject mRNA, the body destroys it immediately. It's a foreign protein, right?
Okay.
So they had to engineer the mRNA to be stable.
Okay.
And when it goes in the cells, to really push and amplify protein production. That they did by changing the mRNA sequences. By the way, mRNA is completely synthetically produced outside,
Mm.
completely. And they coated it in a nanoparticle.
Mm-hmm.
Now, what is this nanoparticle? It's a lipid layer
Mm-hmm.
under which this single strand of messenger RNA sits. It's a nanoparticle, it's very small.
Mm-hmm.
And at least for the Pfizer one, it's got a very fine film of polyethylene glycol around it.
Okay.
And the allergic reactions you're seeing are likely due to polyethylene glycol that's around it.
Okay.
At least that's
Okay.
what the scientific thinking is so far.
Okay.
Now, in order to stabilize the lipid layer, it
Mm-hmm.
has to be kept at a particular temperature.
Okay. So that's where the discussion of the cold
Cold
chain
storage
storage.
come. Now, you can engineer the lipid in such a way that it doesn't require cold chain, but every time you switch one parameter, you give up on the other.
Mm-hmm.
So what does a vaccination
Achieve.
what does achieve, right? So just to finish the story, with mRNA, when you give a nanoparticle, it escapes recognition by the immune system.
Mm-hmm.
Therefore, there is no built-in immunity against mRNA. The immunity comes against the spike that it goes and instructs each cell to produce when it goes into the
Mm-hmm.
cells. So you can give the vaccine repeatedly.
Mm-hmm.
While if you have a vectored vaccine, once immunity against the vector occurs, vaccine is less and less effective.
Aha. Okay.
So you see, there are this is the science of this is really built about balancing all these things and seeing what is the most effective way to do this.
So,
So
Mehul, just one small thing
yeah,
here.
yeah.
Dr. Fauci, I was attending one of Dr. Fauci's talks
Mhm.
at the Johns Hopkins University, and he was saying one of the key mechanisms to make sure that all of this works out stably in the next 2 3 year horizon is to have an adverse event monitoring system, because
Correct.
especially mRNA is so novel a platform that we need to
Mhm.
make sure that we are following and understanding the data that's
Yeah,
coming.
yeah.
Is that why certain countries are a little more cautious about mRNA and then are allowing for these vector vaccines?
See,
Uh,
vectors are also new. It's not that vectors have been there around.
mm,
The best way to think of it is divide vaccines into two buckets. There's the old established technology, there's the new technology.
mm, mm,
The new technology is saying, why are we giving the pathogen? Why are we giving adjuvants? Let's actually do it in a way that the body expands it internally, self-correcting. mRNA goes away in 36 hours. The adenovirus vector is destroyed completely. Body produces it, and we don't even touch the creation of the virus. The old technology or the established technology is take the virus, kill it, take the virus and make a weak strain, take the virus and produce parts of the virus outside and inject those parts. Those are the
mhm,
older technology.
mm,
Those are the established technologies. So, and you must understand the whole science space of vaccination is very conservative.
mm,
They don't like big shifts. But we are moving to the mRNA side and the vectored side because it's a much more efficient way to deliver it. In the first instance, in certain types of vaccines, you can't deliver to immunocompromised patients because what happens if you take a weak vaccine and they're immunocompromised, it becomes a strong vaccine for them and they can get the disease and all that. So this shifts the whole thing away. And so as a platform, mRNA would have become a vaccine platform, it just became faster, right?
mhm,
Now, in terms of Dr. Fauci saying, none of these trials end at phase 3, they
mm,
continue. The other thing people have to realize is that the phase 3 approvals that have come out, at least in the US, everything is under emergency approval right now,
right,
have been approved for the first set of patients. That is, the patients where you needed the protection the most. They're adult,
mm,
right? They 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 may have comorbid conditions, but they're not immunocompromised. They are not children under 16. They are not pregnant women. Now trials for all these subsets are going on. It's not that trials have ended.
mhm,
And after phase 3, there'll be continuous monitoring as an extended phase 3 and also what we call in clinical trials phase 4, which is post market surveillance. You're
right,
looking for adverse events, you're looking for negative outcomes. So, and as you know much better than I do, Chirantan, when you do these multicentric randomized double blind
mm,
clinical
mm,
trials, what are you saying? You're saying statistically my sample that I
mhm,
have taken is a representation of what the broader population is.
right,
And therefore, what I see here will be representative of what I see in the broader population. That's
mhm,
basically the mathematical
mhm,
basis of
mm,
that sort of structure, which means that if you did not see a very, very severe adverse reaction,
mhm,
and they've graded severity, everybody's going to get some vaccine reaction. That's part of vaccination, right? You're
mhm,
going to have a sore arm, you're going to have redness, you're going to feel, headachy, and you see that. Some people didn't see anything. Some people felt, a little sick for a day or two, and they were fine. They were fine. Um, which is what you call reactogenicity. So you need immunogenicity and reactogenicity. So immunogenicity you need high, you need the
mm,
reaction low. That's the optimum vaccine.
mhm,
But as you try and push immunogenicity high, reactogenicity tries to go up. Did you see anything terrible? Now, if you look at the data so far, there was one or two cases of Bell's palsy that occurred, which is really paralysis of the side of the face that reversed. Uh, that occurred in, I think, one of the mRNA vaccines, I think Moderna, but that really reversed and they couldn't make a correlation because that occurs in the normal population also. You must understand that when you do this, what occurs in the normal population
mm,
in your cohort may occur. Pfizer had to stop, not Pfizer, Oxford had to stop trials because they had one case of transverse myelitis where the spinal cord inflammation occurred. And correctly, they stopped trials, and correctly, they are now watching to see is there any correlation. So I think what you're seeing is the following. Pfizer did release its phase 3 interim data.
Right.
They had a wide spectrum. I'm not Pfizer, I apologize. Pfizer released its full data, phase 3. Moderna has released phase 3 data. Oxford released its phase 3 interim data,
Mm-hmm.
at least published. What they've submitted to the regulators, I don't know, because I'm not privy to that. And in that, you're right, that they had a big disparity between their efficacy in Brazil of 64, 65%,
Mm-hmm.
and what they showed when they had this dose uh, issue, right?
Right.
So what happened is that this is what happens when you take vector vaccines. You have to be very tight in production, and vector vaccines you have to make sure that you're testing potency, you're testing dosing, you're testing, because they are viruses, right? So you're taking
Mm-hmm.
that whole production is mRNA is all synthetic. Production is much
Mm-hmm.
faster. It just
Mm-hmm.
in complexity of production. And Serum Institute has done a great job, in terms of their ability to do it. Um, and you have to give them a lot of credit for doing at-risk manufacturing. I mean, it
Mm-hmm.
takes guts to do it
Mm-hmm.
commercially, okay, at this scale. So what happened is when they were looking at a certain batch of doses, they the assessment of the potency suggested the potency was much higher
Mm-hmm.
when it was not. It was really the way the assay was done. There was an assay issue. I think it was one of their production sites in Belgium or Europe somewhere.
Mm.
Um, so they said cut the dose in half.
Right.
So they cut the dose in half. So they ended up actually giving half the dose and the full dose.
And that the half dose had a better
But
response. Right.
so their data showed half dose is a better response. Now, first of all, it wasn't part of the trial.
Mm.
Second, it
Right.
was a subset of patients, right?
Yep. Yep. Yep.
Thirdly, all the patients, I believe, are under 55, and the real
Mm.
risk is patients over. But you do know that when you switch, I mean, this is all research science, right? When you switch the research methodology, you
Yeah.
create
Yeah.
inbuilt errors of conclusion, right? You get
Yeah.
these errors. So I think when the regulators looked at it, the other thing that happened is in some cases, the second dose was supposed to be given after 28 days or somewhere around there. It got delayed to 3 months.
Mm-hmm. Mm-hmm.
So you had these compounding errors going on, and the US said, go and clean up your data and come back. That's basically what the US has said. Give us
Mm-hmm.
a phase 3 data that's cleaner, and they haven't approved it. What the what the British regulators said is, let's look at all the data again. I think they went down and they completely disaggregated the data. They looked at it. They came to the conclusion that uh, it was less of a two-dose, half and full dose issue. This is my understanding. But actually, they could wait because if you see the dose response curves and the risk for COVID incidence, it sort of right the it risk flattens out while the placebo or in their case the other was a meningococcal vaccine that they were their control arm. The control risk goes up for COVID. This goes up for a couple of days because the immune system takes time to respond, like 7 to 12 days, and then it flattens out. And they found that, okay, it's flattening out close to 3 months, so we can live with that, and let's wait for the second dose because of the paucity of doses, because of the dose shortages. So that's what I believe the UK regulators looked at. I don't know in specifically because I have not seen the data. And I believe that's what the regulators in India must have looked at and said that, this data looks, it is efficacious, right? It is efficacious. So
So Mehul, the point is that we believe, but we don't know. That's the issue, I think. And even if you look at, they haven't disclosed who is this committee making the decision and
yeah,
what is the
that
data on
you're
that.
right. I mean, that you have to, I don't know how it happened in India. For
And then
example,
the media
Bharat,
latches on, right? Media
yeah.
finds these odd patient who are potentially given the vaccine without the right side kind of informed consent, and then there are
Mhm,
these hesitancy stuff. That, I mean, the other thing that I'm noticing is that, let's say there are two types of scientists in India. At one end you have Dr. Gagandeep Kang, who has talked about this issue of lack of transparency, and being cautious about taking the vaccine. At the other end, you have Dr. Vijay Raghavan, who is the principal scientific advisor to the PMO, who is saying it's okay to try it out. And then, as for the common person, I'm not a medicine. I teach pharmaceutical economics, but I'm not a medicine person. How? What do I tell
Yeah.
my house, or how, like my maid was asking that how should we deal with it? And I said just be cautious. Let's wait for a little bit and see what data is coming, and then we can think about what to do. So how does the
Yeah.
common man? And my students are
Yeah.
asking as
Prof.
well. How does the common man decide here? That would be nice to discuss a little.
I tell you one thing, I'm fully personally in all my work have been in favor of data transparency. You have to show the data, right? I can see the phase 3 data published in New England Journal of Medicine for Pfizer. I can see it.
Exactly.
You know, I can see the data if I want to on Moderna and what they got a EU approval on, emergency approval on. To be fair, Pfizer published all its phase 3 interim data, and then the final data. Maybe there is a source. Maybe I know Oxford has said this, AstraZeneca said it's completely open, and I believe them because it's Oxford is involved. Oxford is
Mm,
going to do it the right way, so it's all available there. One has to go and parse it out. I do know in case of Bharat Biotech, the data
mm,
that they submitted, it is the pre-pub paper that
mm,
they did with
mm,
ICMR.
mm,
That's a phase 1/2 data set. It's
mm.
around over 300 patients. That's it.
Right.
And then when they did get emergency approval, they must have had more data. I don't know where to look at it.
Mm,
But
mm,
your point is right. If you want to address, see, if you look at the studies on vaccine hesitancy, some hesitancy comes from entrenched beliefs, which is really hard to to bridge.
mm.
Some hesitancy comes because of lack of proper information.
Exactly.
And you need to actually, in things like this, when so much is at stake, I think need to very clearly show the data for what it is, right?
Mm-hmm.
Now, in terms of effectiveness, you know, if you have Brazilian study showing 65, UK study showing this mixed result, and then they must have done most data. Honestly, I have not seen it. Doesn't mean it's not there. I'm not into this space of vaccinology. It's not my
Mm,
expertise,
mm.
but I'm doing a lot of global health work. But it's important that that data it is presented to the population when you're going to vaccinate them. That this is what we saw. This is what the data looks like.
Right.
This is what the efficacy is. And because you're not going to get mRNA in India. It's going to take time.
And that's
And
the other interesting thing, which is the Pfizer approval is in. I mean, the Pfizer submission is in, but it has not yet been approved. So I don't know what's the political economy there as to why that is not being approved, and let
yeah,
the private markets deal with how Pfizer thinks about distributing it at
yeah,
whatever price.
yeah.
Okay, so that this is actually a good time for us to segue to the larger question we started the conversation with. Out of all of this, what do you see the future of clinical trials for biopharmaceuticals and vaccines? How do you see
Yeah,
it
yeah.
shaping
No,
up?
that's why we are doing this talk. I think my larger question is how can you accelerate
Mm,
valuable drug discovery to bedside
right,
process in any country, right?
right, right.
And therefore, clinical trials is a means to an end.
Mm-hmm.
It is about the whole thing. And I really feel you have to start out by, let's talk about India. If India needs to be in the space where it becomes one of the global leaders in getting our drugs, including trials, to the bedside for the Indian population that are meaningful to the diseases that we see as burdens of disease and health, we need to have an ecosystem of discovery, translation research, and clinical research that is really robust.
Mm-hmm.
It's not just regulation.
Mm,
Regulation is just one piece. It includes investment. So, what are the
mm,
different components that are missing? The first component that is missing is really creating a ecosystem of innovation and discovery at the level when you educate the healthcare professionals.
mm-hmm.
We have to have the entire pathway of a physician scientist.
Yes.
Can I do an MBBS, okay, an MD, PhD in India today? Can I get a
Mm,
joint degree? Very hard, right?
mm,
I don't think the MCI has approved dual degrees. Secondly, we need environments of research. Research just doesn't occur.
Mm.
Investments in ecosystems that can do life science research in the, either in the healthcare delivery hospital side or in the medical school side. You have to do it. And I don't want to exclude nursing schools because they also play a very important role.
Mm-hmm.
So you
Mm-hmm.
first have to develop the talent and the expertise and the knowledge to do research at scale.
Mm-hmm.
That has to occur with a system to do research where environments can occur where you can do research, and an investment in supporting research and actually getting research technology or technology that can support research to occur. Let me give
That
you a
is not
case in
happening,
point.
right? That is not
Huh?
happening. That is not
That's
happening.
well, we need it. God, this is something that I have been trying to push. Why is it that we don't have a national genomic strategy?
Mm-hmm.
What is the how did they find the mutations in UK? Because they had the National UK Genomics Consortium.
Mm-hmm.
Surveillance in genomics is very important. Genomics affects all parts of medicine and discovery and science today. Why is it that certain countries in our part of the world have taken global lead in genomics? The Beijing
Right.
Genomics Institute does 30 to 40%
Yeah.
of the world's gene
Yeah.
sequencing.
Yeah.
These are investments made. Can we do CAR T in India as therapy, which is T cell chimeric antigen therapy, which is revolutionary for certain cancers. So we have to invest in the infrastructure. We have to invest in the funding of research. Discovery has to become a path. So, it cannot be that our cohort of life scientists are, considering the size of our country is so small. We have to invest in that because the rewards, the economic rewards are rich. And then we have to have a system where investors can put in risk money or
Right.
early investment cycle money. We
Right.
don't have that ecosystem. Everybody wants to invest when you have margins and markets.
Yep.
Who's going to take from discovery to translation? Who's going to
Yep.
take from translation to clinical? Then you come to clinical, and that also, because I did a lot of work in clinical research. In fact, we set up I was instrumental in setting up the uh, you know, first initial types of site management organizations. Clinical research requires a certain rigor. It requires a certain standard. Most importantly, it requires a certain code of ethics. Now, what it many countries did, and I worked very closely with the person who set up the first Office of Human Research Protection under President Clinton. Dr.
Mm-hmm.
Greg Koski set it up. And what was that all about? Human subject protection.
Right.
Okay? Because this dialogue and narrative can switch very quickly to experimentation if
Mm-hmm.
you don't do it correctly versus benefit. Because if you can bring novel therapies and you can allow them to come in, you can treat different types of cancers, patients will benefit, but they must make sure that it is done with the right ethical standards.
Right.
So, getting the IRB structures very well corrected, getting oversight done, making sure all these things you're saying, informed consent, it has to be informed and it
Mm-hmm.
has to be consent. It
Mm-hmm.
cannot be coercion and ignorance, right? You
Right.
have to have informed consent. And there's a standard you have to adhere to. And getting our clinical research infrastructure in place, why is it that we should be leading in clinical research in the
I
world?
know. I mean, what worries me is that what we should be doing, not only have we not been doing, but even with this kind of shock, I don't see a taste or appetite towards that kind of a direction. Instead, what I see is like Ayurveda doctors will be allowed to do surgeries subject to certain conditions,
Yeah.
and Ayush medicines like being pushed without enough science. I'm nothing against Ayush medicines, but so long as it passes through the trial standards, let's have those standards done, and then we can think about the stuff.
Yeah.
I don't know. I mean, that's my take. Um, Coming back to the question, any like last thoughts on clinical trials and biopharmaceuticals from the India perspective? We will have a primarily India audience here in the podcast. So, any last thoughts on?
I do have. I think, I'd very much like to see a national focus on building India's national healthcare data stack
Mhm.
much more than it is. But it's not only the clinical data stack, It is the biological tissue data stack,
Mm-hmm.
right? It's the genomic data stack.
Mm-hmm.
It is doing predictive analysis on our own population for our own diseases,
Right.
and then figuring out what is the most optimum therapy, making sure that clinical research and drugs that we get are all drugs that are actually tailored to the type of ethnic diaspora we have in terms
Right.
of response curves and all. Because if you see a lot of studies that we a lot of drugs that come out in the market, many of the drugs, significant portion of the trials have been done in Western populations, right?
Exactly.
And I mean, I can tell you there are certain studies that are done in white Caucasian male populations. Now, you tell
Mm-hmm.
me what is the, ethnically, phenotypically, genotypically, we are going to give that drug dosing to a female from South India? I mean,
Mm-hmm.
think of the, where is the corollary? So
So
I think
this,
we,
sorry, I
go
didn't want
ahead.
to interrupt you, but this reminds me of a startup which I think is no longer around, Avestagen, which was
Hmm.
trying to do the Parsi genome and create a data
Yeah,
stack out of
yeah.
it.
You told me that.
Yeah,
Yeah. And
go
that's
ahead,
so
go ahead.
important. I have so many Parsi friends who say I have G6PD, right?
Mm,
I have G6PD deficiency. Can I do this? Can't I do this? I said, why doesn't the Parsi community create its own data stack?
mhm,
Right? How are these Parsi communities? Why is it that they are G6PD? What are the drugs? What are the resource bases? Take it and build it, because we are a heterogeneous country,
yeah,
ethnically, culturally, and we cannot be take a homogeneous paint over the whole country and say this is how it's going to be. There is no, in fact, I do think if we get into this, there is so much power for the rest of the world,
yeah,
economic power we can wield, right? Because we have the computation skills, we have the intellect, we have the educational base, we have the language power, we have the, I would say, legal structures,
mhm.
right? We have to put it together and focus on these things, because these are all engines for growth for the future.
Awesome. Thank you so much, Mehul. I think I really appreciate your time and thoughts. I know you keep a very busy schedule, and we appreciate your thoughts both on the science, on vaccines, and also on how India should create a data stack across the spectrum, like you just mentioned in the last part of your session, to create its own indigenous self-sufficiency in understanding diseases and solving it for the long run. I really appreciate it. Uh, we
Thank you.
will
Thank you, Chirantan.
love to host you sometime in campus once things ease up a
Anytime.
bit. Thank you very much.
Anytime.
Bye
Thanks
bye.
so much for having me. Bye.